go. Yeah, thanks for having me. So I'm gonna talk about what we've been doing to try and understand the immunology underlying some potential HIV vaccine development. For anybody who's not familiar, I'm over here down the street at LJI next to the Moores Cancer Center, along with my other immunology colleagues here. And I'm going to tell a couple of stories that all connect to the same theme, and I'm gonna try and focus mostly on the theme, really centering on why is it so hard to get a good antibody response to HIV envelope primer, these broadly neutralizing antibodies. There's been a lot of interest in potentially having a broadly neutralizing antibody-based HIV vaccine for a number of years now, and the CHOVID and CHOVed HIV Vaccine Consortium, led by Dennis Burton at Scripps. We've been working hard on this problem in different ways, and for me, a lot of this challenge looks like it boils down to this HIV envelope spike structure being so complicated for B cells to recognize. And in part, that's because it has all of these glycans on the surface, which are self-glycans, as well as some other problems. And so to summarize that, where we've gotten to, it looks like this is really predominantly what I consider a B cell immunodominance problem, okay? Where this is the HIV envelope primer, and what happens is, well, it used to be nobody could make the envelope primer until immunizations were with GP120. And immunizations with GP120 didn't elicit, quote unquote, tier 2 neutralizing antibodies, much less broadly neutralizing antibodies. But then this envelope primer definitely has, soluble envelope primer definitely has proper neutralizing epitopes on it. But still, if you immunize monkeys, half the monkeys or most of the monkeys didn't even make tier 2 neutralizing antibodies, much less broadly neutralizing antibodies. And so that was perplexing. And to me, this largely boils down to, again, a problem of B cell immunodominance, which I'm gonna talk through here, where it's these purple spots, for example, are spots where neutralizing antibodies can bind. And so if you have, let's say, monkeys that make neutralizing antibodies, they bind these sites. But those B cell responses are disadvantaged for some reason. And instead, the bottom of the trimer, which is just a much more accessible, quote unquote, easy epitope, dominates the response. And in fact, so we had proposed that as a possible explanation. And now we've had a whole series of studies. Really, I think, well, proving is a strong word, but I think essentially showing that it really is a B cell immunodominance problem, even though at the time we proposed it, that was controversial for a bunch of people. And in fact, we and others have lots of measurements now that indicate that if you immunize with this HIV trimer, something like 99% of the antibody response might be against this base, the bottom of the trimer, which is totally useless because this is the base that's normally covered up by the viral membrane. It's only this structure only exists because it's a soluble protein. You basically cut it off, so to speak, from the surface of the virus, and it's not covered with glycans, and so it's really easy for B cells to recognize. And so you end up with a ton of B cells that recognize this, and essentially none or very few that actually have neutralization capabilities. And that comes down to a competitive problem, which is this immunodominance. And if you immunize with a GP120, the same kind of thing happens. There's this red internal surface here that's totally useless for neutralization, and it's a very easy surface to recognize, so most of the B cells recognize that. We think this is a general problem in immunology. It's not restricted to HIV, but HIV is a fairly extreme example of it, and so we've been trying to understand it both because it's relevant for HIV vaccine design and because it's probably relevant for various other infectious diseases and vaccines. I'll say, for example, SARS-CoV-2 and COVID-19 is not this way at all. And we know that because if you immunize animals with the SARS-CoV-2 spike protein, they'll generate neutralizing antibodies quite quickly, frequently after one immunization. And people infected with SARS-CoV-2 will make neutralizing antibodies to the virus within something like 10 days after symptom onset, which is relatively quick. And then when labs like Dennis Burton's and others have cloned out neutralizing monoclonals and sequenced them, lots of different B cells can neutralize that virus, indicating that it's pretty easy for B cells to, quote unquote, solve the problem, binding to a neutralizing epitope on SARS-CoV-2. And those B cells generally have very few mutations, very little evidence of affinity maturation, again, indicating it's a pretty easy virus to neutralize. The B cells don't have to evolve in germinal centers to get higher affinity to be able to neutralize it, whereas HIV is really very much at the other end of that spectrum. If you've got a situation like this where you've got this protein either on the surface of the virus or in the context of a vaccine, that's incredibly difficult for B cells to end up with a neutralizing antibody response. What's the mechanism for that? Okay. And so one mechanism is you've basically got easy epitopes on a protein and difficult epitopes, and there's a reasonable chance, and definitely the case for HIV, that the difficult epitopes are the ones that are neutralizing epitopes. They've been shielded or made difficult by the virus in some way. Whereas easy epitopes might be like this base or from a denatured protein or something. And so if B cells can bind those easy non-neutralizing epitopes with a better starting affinity, right? If you just take a look at the whole B cell repertoire in a person, and of all the B cells that can bind this viral protein, the ones that bind non-neutralizing epitopes have a much better affinity than the B cells that bind the neutralizing epitopes, and maybe they can just barely hang on, right, to the neutralizing epitopes at time. Well, then the B cells that bind the easy epitope have a competitive advantage because they have a better affinity for the antigen. And so they'll have some potential for immunodominance, for outcompeting the B cells that you actually care about. A second possibility is a numerical disparity, right? So there might just be a lot more B cells that can recognize easy epitopes than difficult epitopes. This happens for HIV, for example. We've two different ways. One, this base is just a large exposed surface that B cells can approach with many different angles of approach, and so there are just going to be lots of solutions to binding that surface. Then on the flip side, most of the neutralizing epitopes are shielded by self-glycans. So any B cell that actually binds human self-glycans, right, has a likelihood of being eliminated during B cell development in the bone marrow because it's self-reactive. And so you're going to have some sort of numerical disparity, and it becomes a question of how big. And then a third scenario could be B cell extrinsic, which is the T cell help in an immune response. To get almost any neutralizing antibody responses, those are T cell dependent responses where the T cells have to provide help to the B cells, and those are specifically the follicular helper T cells that we tend to specialize in. And it's easy to consider a phenomenon where the T cell gets primed and it goes to the TB border, and the B cell gets primed and it goes to the TB border. And this is, let's say 2 days after the infection or immunization. And there's been no proliferation of the B cells and very little proliferation of the T cells. So there's very little T cell help available for the B cells. And so if you imagine easy epitopes and difficult epitopes, right? Well, if there are just a whole bunch more B cells reactive to the easy epitopes, maybe the T cells only help those common B cells, and the rare B cells just get left out from the beginning, which becomes really problematic because after that point, you then have exponential expansion of the immune response. And so any problem that starts out early might get worse and worse, okay? We've shown in a series of studies now that I think this B cell immunodominance does exist. And then what I'm going to mostly talk about today, what I'm going to talk about today is, can that be modulated? Are you just stuck with this problem of if there are lots more B cells to easy epitopes and you really want these, there's just nothing you can do about it? Or can this be manipulated? And certainly one way to manipulate it is by protein engineering, and that's a focus of a lot of fantastic labs. How to change the Hiv Envelope Primer, right, to try and preferentially expose neutralizing epitopes compared to non-neutralizing. And what I'm going to talk about are there immunological ways to manipulate B cell immunodominance? Are there different ways the immune system recruits and recognizes or incorporates B cells into a response, which may be, again, important for various infections and vaccines beyond HIV. So I'm going to talk about these three. And this is what we published last year, and I'm going to cover it in a very brief way. But what we observed is, a normal way to immunize is you have a syringe, right? And you inject, and that's a single bolus immunization. And what we decided was that maybe the immune response would be different if we slowly delivered the vaccine. So same amount of vaccine, same amount of protein and adjuvant, but just over a longer period of time. And we could do that either with slow delivery osmotic pumps, which have been around for like 30 years for drug delivery, or an escalating dose approach where it's still syringe injections, but instead of once, it's several times over a period of days, just again, dividing up the same total dose over time. And what we observed was this makes a dramatic difference. So it's the exact same HIV protein being immunized into monkeys. And if we do that with a conventional immunization strategy, there's no neutralizing antibody response. But now we do it with a slow delivery strategy, and we get a 30-fold increase in the neutralizing antibody titers. And all of the monkeys had detectable titers, and that happened both for this strategy and this strategy. So why does that happen? Well, for one, it's changing the immunodominance. Here's an electron microscopy from Andrew Ward's lab at Scripps, where we see basically what we had predicted, which was after a conventional immunization, almost all of the antibody responses against the base, the bottom of the trimer, which is totally useless. But when we immunized with a slow delivery approach, now we start seeing a lot more diversity of the antibody response against the envelope trimer. Again, same protein, just a change in the immunization strategy. And we think that occurs for several reasons that end up changing the immunodominance overall. One is that we think if you give a bolus injection of a fancy protein, what we've seen by doing a number of studies is that the peak of this B cell response, the peak of this germinal center response, is probably 21 days after immunization. That's a long time from when you first injected the protein. And so probably in those germinal centers, a lot of your favorite protein's been broken up into little bits and pieces by that point. So instead, by having a slow delivery approach, you just have more of your proper protein, okay, available in the germinal centers at the time that there's a peak. We also have data that actually, by that slow delivery mechanism, their rarer B cells get primed better. So you actually recruit a more diverse population of B cells into the germinal centers with a slow delivery strategy. So that changes immunodominance because rarer B cells basically get a fighting chance. They get recruited in this case, and they essentially fail to get recruited in this case. And lastly, by having this slow delivery, we think by changing those kinetics, you actually get more immune complex formation, and therefore more of the protein you inject actually gets retained and becomes productive in the immune response. And this was shown strikingly by Darryl Irvine at MIT, our collaborator on this project, where his lab imaged whole lymph nodes. So these are whole monkey lymph nodes after immunization. And so here's what it looks like after a conventional immunization. There are little, this is a movie, there are little dots rotating here. Almost none of the HIV envelope primer is actually present in that lymph node 2 days after the end of the immunization. But this is what it looks like after either of two different slow delivery strategies. There's just a massively more efficient retention of that antigen, and so then you can get a much more productive immune response. Okay. Now, one doesn't expect to be running human phase 3 clinical trials with escalating dose immunizations or osmotic pumps. And so what's a practical solution to that? Oh, sorry, I just wanted to show one slide that one of the ways we figured this out has been to make use of fine needle aspirates, which is something that I learned from clinician colleagues, but it hadn't been used basically at all in veterinary research. And so we convinced our veterinary colleagues and collaborators to try and do fine needle aspirates to sample germinal centers in these monkeys, and it's worked fantastically well. And now lots of labs are doing this, and it's now starting to be used in human vaccine clinical trials. But what I wanted to move on to was, practically speaking, right? We defined an interesting immunological mechanism, right? That the kinetics of antigen delivery can fundamentally change the immunodominance of the B cell response. But how could we do that in a practical way that may be effective for immunization? And here, a graduate student in Darryl Irvine's lab at MIT came up with this clever idea of trying to change alum. So alum's the most widely used adjuvant, and it has certain biochemical properties. And it turns out that if you have phosphorylated serines, you can get your protein to bind quite well to alum. Most proteins actually come off of alum quickly. And so, and here's an example of this where the animals immunized with a slow delivery approach still had antigen a week later. And so what happens here is usually protein comes off of alum within a matter of minutes, essentially. But instead, these proteins are tightly bound to the alum, and they're being slowly released over a period of something like 14 days, which is basically what we had already shown in mice and monkeys before to be a very effective slow delivery strategy. And so when we do this strategy in mice, we did this to try and put it under a challenging condition like HIV. We know that B cells that can neutralize HIV well are rare, and so we figured that out in humans, that the frequency of those B cells is maybe 1 in a million B cells. So we developed mouse models where the B cells of interest would be rare, present at about 1 in a million, and ask, do those B cells participate in the immune response? And essentially, if we did a conventional immunization, the answer was no. B cells that are rare, present at a 1 in a million frequency, basically get excluded from the immune response. They're too rare. The B cells that manage to participate are probably present at a frequency of like 1 in 10,000. Okay, big separation. But if we now do this slow delivery immunization strategy, this PSARE strategy, now we get this, well, what is it? It goes from 0.1% to 10%. So we get a 100-fold increase in the abundance of B cells with HIV neutralizing capacity, or neutralizing potential, sorry, in these germinal center responses. So we're excited about that vaccine technology, and we think it might have a lot of different applications. So that's one scenario. The next scenario I wanted to talk about was, well, what about T cell help? We spend a lot of time and energy trying to understand CD4 T cells. How do T cells actually fit into B cell and antibody immunodominance? What happens if we add more T cell help, right? So from a vaccine, that's conceivable. You might imagine there'd be ways to prime for T cell help early, so there'd be more T cell help. But the question is, would that be good, bad, or neither? Maybe the T cell help doesn't change anything in terms of the immunodominance, or maybe it makes it worse. It's possible that having more T cell help early just drives the winners to be more winners. It doesn't help the quote unquote losers to participate. Or, maybe it does add more diversity to the response to have early TFH. Right, this is my video of TFH cells and germinal center responses. I'm gonna skip that one today. But we've learned a lot about how early TFH develop and how T cells help B cells over the past decade. But we actually haven't had a good HIV model for that in mice. We didn't have, mice don't make HIV neutralizing antibodies in general, but we've been able to work around that problem. But we didn't have any HIV specific, HIV envelope specific CD4 T cell models. So J.H. Lee and my lab mapped T cell epitopes in GP120. And then she cloned out individual CD4 T cells that could recognize HIV envelope. And then she went and made 2 different T cell receptor transgenic mice. And so now we have an experimental model where we have HIV envelope specific CD4 T cells, which we call HiCap, and we can transfer those into recipient mice and modulate how much T cell help is available for the B cell response and ask, does that affect the outcomes? And sure enough, this is just adding more T cells and showing that, yes, when you add more T cells, you get more T cells. And interestingly, when you add more T cells, you get a lot more rare B cells participating in an immune response. So here, the overall magnitude of the B cell response is going from like 1% to 2%. That's not a very big shift. But the shift in the response of rare B cells goes from essentially 0 to 10 to the 5th, right? So something like a 10,000-fold change in the ability of rare B cells to participate in the germinal center response compared to common B cells. And again, in general, what that translates to is a much better recruitment of B cells that have neutralizing capacity. And in this case, it's actually BNAB, broadly neutralizing antibody capacity B cells. So that's good news. It looks like if we could augment T cell help, that would in fact augment the ability to get not just total B cell responses, but actually better quality or B cells that are responding to rarer epitopes, and so therefore probably more neutralization bias. Okay, and so then the final story, which we just published recently, is what about antigen valency? So in terms of vaccines, vaccine development, there's a ton of interest in multivalent antigens like nanoparticles or virus-like particles, and essentially everybody's pretty much in agreement that multivalency is a good thing, whether it's nanoparticles, VOPs, or liposomal antigens. But there's actually not much immunology to support that, surprisingly. And in part, that's because it's a very difficult problem to study, which I'll explain. And also in part, the outcomes are usually only measured in terms of antibody titer, which can be misleading about which B cells are participating, okay? So we decided to go after this, and we did this in close collaboration with our good friend and colleague, Bill Sheaf over at Scripps, who's just a fantastic protein design expert. And he's designed this EOD construct, which is a germline targeting construct that's now in human clinical trials for HIV broadly neutralizing antibody priming, and fantastic design, clever stuff. And it's actually the 60mer that's in the human clinical trials. But what we wanted to understand was quantitatively, how does valency affect outcomes? So if you have an antigen like this that has 60 copies, how does that compare to an antigen with 1 copy if you immunize with an equivalent amount of protein? What's the difference in the way the B cells see those particles? what's the difference in the magnitude of the response, what's the difference in the qualitative characterization of the response, and does it change, again, sort of immunodominance, rare versus common B cells participating or not. These are really pretty basic immunological questions that haven't been answered, and they definitely directly tie into viral immunology because, you know, viruses look like this, not this, but viruses also have monomeric proteins. So how do these things play out? One of the reasons this hasn't been known is it's really hard to do something like this, to basically try and express the same protein with a series of different valencies. And again, Bill Schief is someone who's quite good at the protein design, and so it was possible to get proteins of a proper construct for this. And this is the EOD that I was just describing, which he's published. I've already described the basic mouse model, although I did it very briefly. Essentially, we adaptively transfer VRCO1 class, which is a BNAB, which is broadly neutralizing HIV broadly neutralizing antibody class. We transfer in transgenic B cells that are specific for this VRCO1. And again, we normally do it so that the mouse ends up having rare B cells, 1 in a million B cells that can actually be of this BNAB class, and then we explore what happens. And so this is all done in this study by postdoc Alex Cato in my lab, who has a lot of talents, and one of them is intravital microscopy. So this is live microscopy of a mouse lymph node, and the green cells are the VRCO1 class B cells. So those are the HIV specific B cells, which in this case have been transferred in large numbers so that we can actually visualize them. And then the blue B cells are non-specific B cells. And you can see they're all cruising around the lymph node doing what they're supposed to do, okay? So now Alex immunizes with this 60-mer. And what happens is that the B cells that are HIV specific basically stop migrating. And so that's a classic indicator of a B cell that's become activated. You see the blue B cells, basically, they're just all still cruising around. But the green B cells have seen something, and you can tell that they've seen the antigen because they basically stop. And what's intriguing is if we now do that with monovalent antigen, it doesn't do anything. So here's monovalent antigen, and here's the 60-mer. So these B cells stop, these B cells don't. So essentially, the B cells are blind to monomer, which means that when you go, if someone goes and injects with GP120, right, which is really the classic HIV antigen, the B cells really don't even see it, which is probably one good reason that people and animals immunized with GP120 really make lousy, or in general, no neutralizing antibody responses, because they're really, those monomers really aren't activating B cells. And we can quantify all of this, and there's a gradient. There's no activation with a monomer. There is some B cell recognition of tetramers and octamers, okay? And in fact, that initial activation translates into a lot more proliferation of the B cells that see the multivalent antigen, and also a better recruitment overall of the B cells. So for example, if the mouse is immunized with a 60-mer, essentially all of the B cells get recruited into the immune response. But if the mouse is immunized with a 4-mer or an 8-mer, that does activate some B cells, but it ends up only activating about half the B cells. About half the B cells just sit there and don't do anything, which is pretty intriguing. Whereas with the monomer, nothing happens within this period of time. And it also drives changes in migration markers and whatnot. And so we could then go and check. We say, oh, well, so maybe when the B cells get activated with a high valency antigen, maybe that enhances the immune response, both by just direct activation of the B cell and driving the B cells to seek out more T cell help at the TB border. And so we could do that with whole lymph node imaging. The white cells are the B cells, and here they're just all scattered everywhere. And you can see after immunization with a 60-mer, all of a sudden within, I think it's 24 hours, they're driven to the TB border and they're very strongly clustering. So a very big difference in the behavior of the cells depending on the valency of the antigen they were exposed to. All right. And so the other thing we wanted to look at was then, okay, the valency is affecting the B cells, does it change the way the B cells interact with the T cells? If the B cells see something that looks more like a virus, something that's multivalent, does that change the interaction with the T cells or not? And if so, how does it do so? And this was a lot of work for Alex Cato, but he did intravital microscopy of these HIV-specific B cells and antigen-specific T cells that were transferred in. So the antigen-specific T cells are in pink and the B cells are in green. And here's when they're immunizing with a 60-mer. And you can see after this that they're making these huge clusters of B cells and T cells just within 24 hours of seeing the antigen. And this is quantitation of this. So every line is a different cell, a different B cell. And red boxes are when it's interacting with a T cell. So lots of interaction between the B cells and T cells and for long periods of time. And so in contrast, here's a mouse that's immunized with a tetramer. So it's still a multimerized antigen and it's the same amount of protein. It's the exact same specificity. But now when the B cells see that, there's almost no interaction with the T cells. So a really dramatic difference in B cell T cell interaction based on the valency of the antigen. Interestingly, if we now do the same thing with a low affinity antigen, so we've got different engineered versions of these antigens. So now here's an antigen with a 50-fold lower affinity. So that's, basically that's a much lower affinity. And we've shown this is selected based on things that we found in humans, so we know this is a physiologically relevant range. Both what we call high affinity and low affinity are both affinities of interest. And so if we immunize with a 60-mer, a large multimer, but it's 50-fold lower affinity, it looks almost the same as the high affinity, okay? Which basically tells us that valency, essentially valency trumps affinity in this scenario, okay? That if you have a highly valent antigen, it's quite good at recruiting low affinity B cells, which was interesting. So overall, from that study, we could conclude that valency is an important factor and it really influences B cells instantly, right? It's doing so within an hour of the immunization. We're seeing differences in the behavior, or 2 hours. We're seeing differences in the behavior of the B cell, whether it's seeing just a conventional monomeric antigen or if it's seeing a multivalent antigen. And really, B cells fail to respond to monomer at physiological affinities. And really, I think the way B cells end up responding to monomer is that they just take up a little bit of the monomer. It doesn't do anything to the B cells, but the B cells will present enough peptide MHC that if you've got enough of a T cell response, you can finally prime some of the B cells to do something. But really, the monomer is not doing anything to the B cell itself. And that's why, highly valent antigens have such a benefit. In data that I'm not showing, well, no, I showed, I didn't really, I showed this one, that high valency antigens recruit B cells with a broad range of affinities. Interestingly, low valency antigens, like the tetramer, preferentially recruit high affinity B cells. So we don't think that just more valency is always better. It's actually, if you're just trying to get high affinity B cells and you know that your antigen's pretty good, a tetramer could potentially be better than a 60-mer because you wouldn't recruit as many low affinity B cells. But in germline targeting approaches and whatnot, where you're really usually struggling to try and recruit rare and low affinity B cells, a high valency probably gives a huge benefit to the vaccination strategy. And so we've looked at this in a couple of different ways. The rarer the B cells, the harder it is to prime them and get them to respond, and because of this immunodominance problem. In terms of valency, the higher the valency, the better off you are at recruiting rare B cells or lower affinity B cells, and there's the affinity piece. And I think we've really started to give people a framework for how to think about these, to understand these problems immunologically so that they can be applied in vaccine strategies for if you're trying to elicit a B cell response to a particular virus and you're trying to get, some sort of neutralizing response, well, do people actually have B cells that can do that? And how rare are those B cells to this particular epitope versus that particular epitope, which sort of sets the stage for what type of vaccination strategy might you have to do, right, to get a good response. So for example, right, for SARS-CoV-2, it's clear that the precursor B cells to make neutralizing responses to SARS-CoV-2 are totally common. There are lots and lots of B cells that can do this. So you can basically just throw all of this out the window. That's just not a limiting factor for trying to get a neutralizing antibody response to that virus. And so that allows you to consider, a variety of different vaccine approaches. Whereas with HIV and probably with some other viruses as well, if there's rare precursor frequencies, you really need to consider that vaccination strategy very carefully and what types of affinities and avidities might allow you to recruit those B cells and get them to affinity mature in germinal centers. And I talked about that T cell help can also be a big factor, particularly if you've got rare B cells. And so having vaccine strategies that are better at eliciting early T cell help rapidly, either by having more T cell epitopes or priming the T cells better, look like that's probably a really big deal for differentiating between what might be a successful vaccine strategy and not. Yeah, I think I will stop there. Here's a picture of my lab at LJI. A bunch of people contributed to this. Bob Abbott set up the initial mouse model. The T cell work that I just talked about was all done by J.H. Lee, who's a fantastic postdoc in the lab, and all the B cell multivalency study and the intravital microscopy and whatnot, that was all done by Alex Kato, Yukato in my group. And so here's a photo of him getting married this past year. And all of the protein designs came from Bill Sheaf at Scripps, and he's been a fantastic collaborator. Couldn't have done any of this without him. And I described the contributions from the collaborations with Daryl Irvine's group at MIT. And everything that I described has been very collaborative research with all kinds of PIs throughout our HIV Vaccine Consortium, which is now called the CHOVD. And many of those people are shown here. So I will, I will stop there, and I'd be happy to take any questions.
Yeah, hi, Shane. This is Doug. I think those are great stories. Thank you. That was terrific.
Thanks, Doug.
So I'm still trying to get my head around the mechanism here. And do these multivalent antigens work better because each B cell has multiple receptors to engage multiple times rather than just once, or is there something else that is going on?
Yeah, good question, and I should have talked through that some more. That's I think it's two main things. So a given B cell has, let's say, something like 10,000 B cell receptors on the surface, and the way it's going to recognize antigen is by cross-linking those receptors. And so if you've actually just got a monomeric antigen, theoretically, you can't activate a B cell because the monomer just binds to one BCR, and there's no cross-linking event, so there's no signaling to the B cell. Now, in reality, it gets more complicated than that because that monomer could just get bound to some surface or other things. But we're going to focus on the other, and we tested that, and it really didn't do anything. So now when you've got a multimer, you will cross-link more BCRs, but also with more density probably, right? So probably the density of cross-linking has some effect. And then beyond that, now when that B cells become activated, well, let me back that sentence up. So it looks like by binding a 6-dimer with greater avidity, right? So there are more BCRs binding this. it looks like that makes it so that the B cell will actually take up more of this protein than this protein, right? Just in a straight microgram quantity, by being more likely for this to trigger an actual endocytic event by the BCRs, and therefore result in that B cell presenting peptide MHC, right, back on its surface that's going to be HIV specific. And these B cells are going to need T cell help. And so B cells that are seeing multivalent antigens, we can infer from this, are taking up a lot more protein and presenting it back to the T cells, such that when they come in contact with the T cell, they now have these very long interactions with the T cells. This is probably reflecting the abundance of peptide MHC that they actually had on their surface. So these B cells and these B cells got exposed to the same amount of protein, but when it was a tetramer, probably these B cells just took up a lot less of that protein and therefore have less peptide MHC on their surface. We don't formally show that because what we also see is that when those B cells that came in contact with a multivalent antigen, I mentioned that they change their migration, but they also express a bunch of other activation molecules on their surface. And so probably it's a combination of things where they are presenting more peptide MHC on their surface, but they're also now presenting, they have also got more just co-stimulatory molecules on their surface. So they're literally just binding to the T cells better, right? Even in peptide MHC independent ways, which is probably just built into the programming of the immune system over a long-term evolution, right? Being programmed to try and recognize more strongly to multivalent antigens, right, as being more threatening. And so not only the B cell presenting more peptide MHC, but expressing more co-stimulatory molecules and migrating to the TB border more as a response. Is that your question?
Yeah, it is. I wonder if I can ask a second one while I've got you.
Yeah.
So getting to SARS, do you think that a multivalent antigen like the Novavax vaccine is more likely to successfully elicit neutralizing antibody than an RNA vaccine or a vector-expressing vaccine of monomers, or is SARS less compromised than HIV or some other viruses in terms of eliciting these antibodies?
Right, good question. So I think it's I think our data indicate it's probably something in the range of sort of an 8-mer, a little higher than an 8-mer, probably. The Novavax one is in the range of an 8-mer. I think what our data indicate is really how bad it is to go with a monomer, essentially there, right? That you're just failing to get cross-linking of the B cells. You're not engaging the fundamental biology of the B cell. The B cell is designed to recognize pathogens, and pathogens normally have some sort of multimer on their surface, and that's probably essentially an intuitive way for the immune system to try and enrich for antibody responses that are functionally useful, right? Because they're presented in some multivalent way on the surface of a pathogen, and you just lose that with a monomer. You can overcome it with a monomer by having lots of adjuvant, and T cell help, which will come up over time. But having any multivalency at all at least engages that basic component of the B cell biology, and so it's going to recruit more B cells and get more efficient neutralizing antibody responses. So yeah, I definitely think that approach by Novavax is better than anything that's going to give a monovalent approach. Now, referring to other SARS-2 vaccine strategies, so let's say the RNA vaccines, those are presenting, I think all the ones in phase 3 are presenting membrane-bound, right, SARS-2 spike protein on the surface of the RNA, essentially transfected cells, right? So that potentially then is a multivalent scenario, right? But it'd be on a planar surface, right, where it's Essentially what you've got there is the surface of the cell, and then you've got spike trimers on the surface of the cell, and B cells would recognize that, and so then you get your multivalency because you've got membrane bound. That's a little hard to figure out in vivo, right? How that plays out, because this isn't in a lymph node, so it's not where the B cells are. So it's not entirely clear in what form the B cells are going to see mRNA presented antigen or vector presented antigen. Maybe it ends up on myeloid cells that migrate into the lymph node, right? And then you have that presentation, but it hasn't been defined. I've seen in some cases people have had viral vectors that have expressed soluble RBD or spike, and those have generally been less immunogenic, which I think is connected to these immunological principles we were just showing.
Great, thank you.
I lost my cursor, but so I can't go to, oh, there we go. All right, anybody else? We're good?
Well, that was great, Shane, thanks.
Sure, thanks for having me.
Super fun. Thanks.
All right,
Have a
have
good
a good weekend.
Happy Halloween.
You too.